Okay, good evening everyone. So I'll share my screen for today. You can let me know if it's visible and if I'm audible. One second. Let me share my screen. Okay. Yeah. So I hope my screen is visible. Good evening. Today is the 8th live session of these live classes of this course, RNA Biology. In these live sessions, as you know, we discuss some previous year assignment questions so that you don't only not only discuss the question, but you also discuss the concept behind those questions, so it's easier for us to discuss similar type of other questions. Okay. So today is the 8th live session for that. I hope my screen is visible. We'll move on to the first question. For today. Nucleoproteins are important in RNA export. True or false? So I'll wait for 4 or 5 minutes. I'll let you figure out the answer on your own. If not, then we'll discuss the concept behind the question. So anyone? Okay, so the answer will be true. So we'll discuss what are nucleoporins and why are they important in RNA transport, right? So let's discuss what are nucleoporins first, right? So if you talk about nucleoporins, basically they are a family of proteins, right? That form the building blocks of nuclear pore complexes, which are large channels spanning the nuclear envelope and mediate the transport of molecules between the nucleus and cytoplasm. So they are the proteins that make up the nuclear pore complex, a massive structure that spans the nuclear envelope. They are essential for nucleocytoplasmic transport, allowing the passage of molecules like proteins and RNA between the nucleus and cytoplasm. They are composed of approximately 30 different nucleoporins, NPCs, or the nuclear pore complex, each with its specific localization and function within the complex. They are crucial for cell function, and mutations in NUP genes have been linked to various human diseases, including neoplastic and non-neoplastic conditions. They have a dynamic nature, like some nucleoporins can be mobile and shuttle between the NPC and other locations in the cell, participating in other cellular processes beyond transport. So alterations in nucleoporin function can even lead to diseases, right? And research into nucleoporins and NPCs is ongoing to understand their role in health and disease. So basically they are a family of proteins which are the constituent building blocks of the nuclear pore complex. And the nuclear pore complex is a massive structure embedded in the nuclear envelope at sites where the inner and the outer nuclear membranes fuse, forming a gateway that regulates the flow of macromolecules between the cell nucleus and the cytoplasm. And nuclear pores enable passive and facilitated transport of molecules across the nuclear envelope. There are a family of around 30 proteins, the main components of the nuclear pore complex in eukaryotic cells. Nuclear protein 62 is the most abundant member of this family. They are able to transport molecules across the nuclear envelope at a very high rate. A single NPC is able to transport 60,000 protein molecules across the nuclear envelope every minute, right? So structure-wise, they aggregate to form a nuclear pore complex, an octagonal ring that traverses the nuclear envelope. And the ring consists of 8 scaffold subcomplexes with 2 structural layers of COP2-like coating, sandwiching some proteins that line the pore. From the cytoplasm to the nucleoplasm, the 3 layers of the ring complex is named the cytoplasm, inner pore, and the nucleoplasm rings respectively. Different sets of proteins sit on either ring, and some transmembrane proteins anchor the assembly to the lipid bilayer. Now, in a scaffold subcomplex, both the cytoplasm and the nucleoplasm rings are made up of Y complexes, a protein complex built out of, among others, NUP133 and NUP107. So, on each end of each of the 8 scaffolds are the 2 Y complexes, adding up to 32 complexes per pore. So, the relationship of the membrane curvature of a nuclear pore with Y complexes can be seen as analogous to the budding formation of the COP2 coated vesicle. So, the proteins lining the inner pore make up the NUP62 complex. So, on the nucleoplasm side, extra proteins associated with the ring form the nuclear basket, a complex capable of tethering the nuclear protein to the nuclear lamina and even to the specific parts of the genome. The cytoplasmic end is less elaborate with 8 filaments projecting into the cytoplasm, and they don't seem to have a role in nuclear import. So if we start from nucleus, we all know what a nucleus or nucleolus is. They are double membrane nuclear envelope with nuclear pores, chromatin and nucleolus. and they store hereditary information like DNA, makes RNA which makes proteins, and makes ribosomes as well. The nucleolus. These are present in both eukaryotic plants and animal cells. Now we know about the rough endoplasmic reticulum. A structure internal membrane is a system of folded sacs and tunnels, has ribosomes on it. That's why it's called the rough. The function is intercellular highway for molecules to move around and out of the cell and makes protein and other cell components. Cell type can be both plants and animals in eukaryotes. For smooth ER, the internal membrane of systems of folded sacs and tunnels, no ribosomes. The function will be intracellular highway, storage area for proteins. Cell type can be both plants and animal cells. Moving on to ribosome, the structure is made up of protein and RNA. No membrane made in nucleolus. And the location of protein synthesis, that's why it's so important. It's called the protein synthesis synthetic factory. Free ribosomes make proteins used by the cell, and ribosomes in ER make proteins for export to other cells, right? Cell type can be both prokaryotic and eukaryotic, like plant cells, animal cells, because proteins are needed in both prokaryotes and eukaryotes, right? Now, if you go to the Golgi apparatus, the structure is stack of membranes or sacs filled with fluid. They functionally processes packages and secretes materials made in the endoplasmic reticulum. And cell type can be both plants and animals. If we talk about mitochondria, which is the powerhouse of the cell, they have double membrane, have their own DNA, have numerous in high requirement cells, muscles, right? And the function is powerhouse of the cell, transfers chemical energy into ATP to be used as an energy for life functions. Now for chloroplast, structured double membrane structure, it contains DNA, contains green pigment which is also known as the chlorophyll. The function will be responsible for photosynthesis, conversion of sunlight into chemical energy, and cell type plants and animals. Sorry, plant cells, not animal cells. Now we come to lysosomes. Lysosomes are structurally spherical organelles, contain enzymes. They help in the digestion of organic molecules, old organelles, foreign substances, and cell types are these. Now, if we talk about cytoplasm, aka cytosol, structure is fluid between cell membranes and organelles, contains water, salts, organic compounds. They help in movement and they can be found in different cell types. If we talk about cytoskeleton, structure network of long protein strands located in cytosol, no membranes, and this supports cell by giving shape and organization, help with the movement of organelles in the cytosol and cell movement. Okay, cell types can be both plants and animals. Cell membrane is a lipid bilayer with carbs and proteins, selectively permeable, boundary and support for cell. Prokaryotic cells, eukaryotic cells, both animals and plants. Cell 1 is present only in plants. It's a wall outside the membrane made of cellulose, has pores, helping protection and support. In eukaryotic cells, it's present only in plant cells and prokaryotic cells. Some prokaryotic cells is also present. Then we talk about vacuoles, which are membrane-bound fluid-filled organelles. They store water, proteins, carbs, and salts. In plant cells, these are large. In animal cells, these are just very small. Now, if we come to the nuclear transport, the nucleus contains most of the cell's DNA, allowing for the sophisticated regulation of gene expression. And the nuclear envelope is a double membrane that surrounds the nucleus. Right? Now, the nucleus contains subcompartments that are not membrane bounded. The nuclear envelope contains pores used for importing proteins into the nucleus and exporting RNAs and proteins from the nucleus. So nuclei range in size from about 1 micron to more than 10 micron in diameter, and most cells have a single nucleus, but some cells contain multiple nuclei and a few cell types lack nuclei. The percentage of the genome that is heterochromatin varies among cells and increases as cells become more differentiated. Now, although the nucleus lacks internal membranes, some nuclei are highly organized and contain many subcompartments. Each chromosome occupies a distinct region or territory, and this prevents chromosomes from becoming entangled with one another. Now, the nucleus contains both chromosome domains and interchromosomal regions. Nuclear subcompartments are not membrane bounded. RNA is synthesized and into and ribosomal subunits are assembled into the nucleolus. The nucleolus containing DNA that encodes RNA and that is present in all the multiple chromosomes. Now, what are mRNA splicing factors? These are stored in nucleus vesicles. They move to sites of transcription where they function. Other nuclear bodies can be identified with antibodies, but the functions of most of these are unknown. So we have the nuclear envelope, the nucleolus matrix, and the chromatins. So the nucleus is surrounded by a nuclear envelope consisting of two complete membranes, and the outer nuclear membrane is continuous with the membranes of the endoplasmic reticulum, and the lumen of the nuclear envelope is continuous with the lumen of the ER. So the nuclear envelope contains numerous NPCs or nuclear pore complex. They are the only channels for transport of the of the molecules and macromolecules between the nucleus and the cytoplasm. Now, the nuclear lamina is constructed of intermediate filament proteins called lamins. And the nuclear lamina is located beneath the inner nuclear membrane. They are physically connected by lamina-associated integral membrane proteins. So the nuclear lamina plays a role in the nuclear envelope assembly and may provide a physical support for the nuclear envelope. The proteins connect the nuclear lamina to chromatins. This may allow the nuclear lamina to organize DNA replication and transcription. And yeast and some other unicellular eukaryotes lack a nuclear lamina. Large molecules are actively transferred within the nucleus and the cytoplasm. Uncharged molecules smaller than 100 Dalton can pass through the membranes of the nuclear envelope. Molecules and macromolecules larger than 100 Daltons across the nuclear envelope by moving through NPCs. Now, large particles up to 9 nanometer in diameter corresponding to globular proteins up to 40 kDa can pass through the NPCs by massive diffusion. Larger macromolecules are actively transported through NPCs and must contain specific information in order to transport it. And if you talk about the nuclear pore complexes or NPCs, the symmetrical structures that are found in sites where the inner and the outer membrane are fused. And each NPC is hematized mass of around 120 to 160 Daltons and is constructed from multiple copies of 30 proteins. Approximately 30 proteins. So it replicates the extended cytoplasm and NPCs and a basket-like structure that extends into the nucleus. So protein organization and nuclear protein complex. Now, the proteins and NPCs are called nucleoproteins. Many nucleoproteins contain repeats or short sequences which are thought to interact with transport factors during the transport. Some nucleoproteins are transmembrane proteins that are thought to anchor NPCs in the nuclear envelope. All of the electrodes of yeast NPCs have been identified. NPCs are assembled and disassembled during mitosis, and some nucleoproteins are dynamic. They rapidly exchange each other from NPC. Now, mature DNA nuclear proteins contain sequence information required for their nuclear localization. Proteins separately enter and exit the nucleus to nuclear pores. Information for certain nuclear inputs lies in the small portion of the transported protein. The receptors for nuclear transport are cytoplasmic proteins that bind to the NLS of the cargo proteins. The nuclear envelope receptors are part of a family of proteins known as carriopherins. So, short stages of amino acids rich in leucine act as the carbon expedient and nuclear export sequences. A nuclear export receptor binds proteins that contain nuclear export sequences in the nucleus and transfers behind them to the cytoplasm. The RAN GTPase controls the direction of the nuclear transport. The RAN is a small GTPase that is common to all eukaryotes. It is found in the male in cytoplasm nucleus. So RAN-GAP promotes hydrolysis of GTP by RNA. RAN-GAP promotes exchange of GTP for GTP on RNA. So RAN gradient between the cytoplasm and the nucleoplasm. In the cytoplasm, the energy needed to create this gradient is supplied by ATP, transforming the RAN-GTP in RAN-GTP. This is the nucleoplasm of the sink of RAN-GTP and a source of RAN-GTP in the cytoplasm, and RAN-GTP is converted into RAN-GTP. Thus, the cytoplasm is a source of RAN-GTP and a sink of RAN-GTP. This way, two gradients are created, RAN-GTP and RAN-GTP. The size icons in the figure depicts level of oxidation. This is a molecular trafficking across the nuclear pore complex mediated by carriopherins, importance and exportins. So the RAN-GAP is cytoplasmic, whereas the RAN-GEP is loaded in the nucleus, and RAM controls nuclear transport by binding carriopherins and affect the stability beyond their cage. mRNAs, tRNAs and ribosomal subunits produced in synthesis are exported through NPCs in function during the transformation of the cytoplasm. Multiple classes of RNA are exported from the nucleus. From mRNAs, tRNAs and ribosomal subunits produced in its nucleus, exported through the NPCs to function during translation in the cytoplasm. Chromatin, the packaging of chromosomes and the epigenome is regulator of nuclear processes. The chromosomes take part in most of the space in the eukaryotic nucleus is bound by ribosomes or chromatin. The DNA goes with DNA proteins to unfold it during the N1000 fold, and the chromosomes are highly even more lightly weighted composition, so show more discussed and heterosomes. DNA is wrapped around the instrument around 4 billion years ago. In the nucleosomes, in turn, are further packaging precision by H1 to form the NT-NM fiber. Cytoplasm used to watch. The 30 nm fiber is further packaged with non-existent protein. Chromosomes have different regions with different functions. Intrachromatin densely packed, telomeres, centromeres, silent regions. Intrachromatin less densely packed regions activate transported genes. So other proteins are histones, H1, non-histone proteins like HMG box, specific proteins like histone reaction, region, etc. The process of the nucleus happening in replication, transcription, RNA processing, recombination, DNA repair, and export of RNAs, import of protein. Now, if we could talk about 25 histone modifications, 25% of the N-terminal part of the histones are not in the rod-term region. N-terminal calls are modeled by acetylation, methylation, phosphorylation, and uridation and ribosylation. Modifications located in the heterochromatin methylation of H3 lysine 9H3K27 modified in actively transcribed regions. So, gene switched on active chromosomes, chromatin, unmethylated cytosines, cylated histones. Gene switched off silent chromatin, methylated cytosines, and the red one, deacylated histones. So, transportation between cytoplasm and cytoplasm is critical for the survival of cells. It is a highly integrated process. Several million macromolecules relatively pass between nucleus and cytoplasm every day. Minute. The nuclear envelope is a physical barrier which regulates the traffic between cytoplasm and cytoplasm. It's a phospholipid bilayer membrane which consists of two areas, inner and outer. Inner and outer membranes are separated by the nuclear space. The cytoplasm is connected to the cytoplasm via nuclear pores. Although small sizes of molecules freely move through the nuclear pore, bigger molecules need the help of a social carrier protein. If we talk about nuclear transport cycle, transportation of the macromolecule including protein or RNAs between cytoplasm and nucleus occurs through nuclear pore complex. It's highly selective and bidirectional. Transporter for various cargo molecules. There are four different factors. NPCs and nuclear proteins, nuclear localization signals, and nuclear export signals. RAN proteins that allows for active transport. The different that recognize RNA molecules. So NPC is a complex vascular structure with huge molecular mass of 120 around kDa. And it is made up of NPS. Each NP has a central role that connects between the cytoplasm and the nucleoplasm. Main features of the NPS include the inner pores ring, nuclear cytoplasmic rings, the nuclear basket, and the cytoplasmic basket. NPC is composed of multiple copies of 30 to 50 different NUPs and 50 to 100 different NPCs integrated into the NPC structure. NUPs serve as an architectural scaffold and permeability conductor. Regulators. Some NUPs have intrinsically disordered domains rich in repeating amino acid sequences. These reporting sequences line the central hole and they regulate the passage of the cargo molecule. So, what is the analysis nuclear localization signal? The proteins acquired for nuclear phosphorus, DNA, polymers, RNA, etc. are targeted to the nucleus by specific amino acid sequences called analysis. Analysis recognizes the nuclear transporter that direct the venue of the cytoplasm through the NPCs. The first NPC was characterized by Alan Smith and colleagues in 1984 from simian virus. It antigen as a classical monoparticle serum PKKKK RV. So the bipartite and NLVs have two clusters of missing amino acids. The prototypical bipartite analysis composed of Sinus larvae. Sometimes proteins remain within the nucleus following their import from the cytoplasm, but many others shuffle back and forth between the cytoplasm. Proteins are targeted for export from the nucleus by specific amino acid sequences called nuclear export signals. The next contains the hydrophobic amino acids that consists of a sequence for NAS is recognized by transporters within the nucleus called the exportin, called the transport into the C of the cytoplasm, and different exportins have their specific cargo molecules. For example, C has exportin to transpose UADA alpha. Cardiopharynx, also known as important cell exotics, are superfamily of real introduction receptors that facilitate the translocation of proteins, RNAs, and nucleic particles across the NPC. More than 20 bypass have been reported among 11 times 15 times ball right. The 15 either flight exportins, 8 exportins are involved in the growth of cargo molecules. Each cardiopharin is its own specific cargo molecule. For example, exportin to control the export of important alpha and exporting. And it is also explore nuclear motif including tRNA, miRNA, RNA, viral RNA, and small nuclear RNA. However, during structural events is highly likely inside the adrenals so that they can be at purpose in the main room. Into ribonuclear proteins. The activity of cardiopharynx are regulated by interaction of a protein called RAN. RAN gives the nuclear transport directionality by through into the nuclear pore and is a GDP protein which concerning evidence is regulated by RNA binding and privileges. And RAN GTP is about 10-fold, 100-fold more abundant life in the nucleosome than in the cytoplasm, which is possible due to the fact that now in exchange from cytoplasm located in this nucleoplasm. Similarly, the rate ATPase activating protein is located in cytoplasm. The activity of GAF and RAN-GA ensures the unequal distribution of RAN-GTP across the nuclear envelope with high frequency of RAN-GTP in the nucleus to provide directionality of transport across the nuclear envelope. So cargoes containing LEAs are recognized by exportins. The NEAs bound exportin from the complex with RAN-GTP. RAN-GTP is 100 times more abundant in the nucleoplasm than cytoplasm. Binding of RAN-GTP to exportins increases the high affinity of exportin for the cargo molecule. The exportin cargo RAN-GTP complex binds the blocking area of NPC and then passes to the nuclear pore. RAN gap is the cytoplasm hydrolysis RAN-GTP cytoplasm while deletion is the destruction of the complex. The transport of proteins to the nucleus is highly regulated event such as transcription factors import and export into the nucleus is a well-established mechanism to control these protein factors. In monster experiment, PFs associated with cytoplasmic protein that mask their NLS and therefore their image cytoplasm. For example, under unstimulated condition, NF kappa B binds with I kappa B cytoplasm, A and I kappa B mask NF kappa B NLS and it remains in the cytoplasm. In simulated condition, I kappa B is phosphorylated and degraded, allowing NF kappa B to enter nucleus and activate transcription and target genes. Another example of transcription factor PHO4 whose NLS is masked by phosphorylation. When this is required inside the nucleus, the dephosphorylation happens is imported to the nucleus. So all RNAs are transported across the nuclear envelope as RNPs. The importance of exportins and RNA dependent manner. RNA is associated with browser proteins and other factors in nucleus exported to this. Then RNA exposed party precursors to exporting Bombay and Bombay Martin. mRNAs are explored by distinct rapid mechanism that does not involve cardiopharynx and is cytosol by membrane. PMRNAs are associated exposure candies in the presence of membrane. So many small RNA such as cell RNAs and RNAs pushes nucleus as a component of RNA processing machinery. Initially, transportase is right to cytoplasm where the big processes to form SNPs and then returns to nucleus. CRM1 and other protein receptor protein that bind 7 prime to 7 7 methyl guanosine capsaicin is involved in the export of SNR cytoplasm. In contrast, sequences present on the SNP proteins involved are in the cytoplasm of SNR cytoplasm to the nucleus. So coming back to the question we started with nucleoproteins, so it's important in RNA transport. Obviously, yes, it is very important in RNA transport. If you have any question regarding this, you can ask me now, or we'll take a break for 4 minutes and then we'll go on to the next question for today. So the next question for today. So the next question is regarding RNA interference. I think someone's mic is on. Can you just switch off the mic if that's not a problem? Okay, so the next question is on RNA interference. So RNA interference was first discovered in zebrafish, C. elegans, Saccharomyces cerevisiae, or Saccharomyces pompi. I'll wait for 2-3 minutes, then we'll go on to discuss. If anybody knows the answer, you can write in the chat box and let me know. Anybody? Okay, so the answer will be C. elegans. Okay, so what was the first detailed look at this? So scientists have gotten their first detailed look at the molecular structure of an enzyme that nature has been using for eons to help silence unwanted genetic messages. So it was seen that a team of researchers with Berkeley Lab, the University of California, Berkeley, used extracrystallography at ALS beam lines 8.2.1 and 8.2.2 to determine the crystal structure of dicer, which is an enzyme that plays a critical role in a process known as RNA interference. The dicer enzyme is able to snip a double-stranded form of RNA into segments that can attach themselves to genes and block their activity. Right? So, with this crystal structure, the researchers learned that dicer serves as a molecular ruler with a clamp at one end and a cleaver at the other and a set distance away that produces RNA fragments of an ideal size for gene silencing. So it has RNA has long been known as a multi-purpose biological workhorse, right? Responsible for carrying DNA's genetic messages out from the nucleus of living cell and using those messages to make specific proteins in a cell cytoplasm. So in 1998, however, scientists discovered that RNA can also block the synthesis of proteins from some of those genetic messages, And this gene silencing process is called RNA interference, and it starts when a double-stranded segment of RNA encounters the enzyme dicer. So dicer cleaves double-stranded RNA into smaller fragments called short interfering RNAs and microRNAs. So dicer then helps load these fragments onto a large multiprotein complex called RISC for RNA-induced silencing complex. RISC can seek out and capture messenger RNA molecules with the base sequence complementary to that of its siRNA or miRNA that serves to either destroy the genetic message carried by the mRNA outright or else block the subsequent synthesis of a protein. Until now, it has not been known how dicer is able to recognize double-stranded RNA and cleave those molecules into products with lengths that are exactly what is needed to silence specific genes. So the Berkeley researchers were able to purify and crystallize a dicer enzyme from Giardia intestinalis, a one-celled microscopic parasite that can infect the intestines of humans and animals. So this dicer enzyme Giardia is identical to the code of a dicer enzyme in higher eukaryotes, including humans, that leaves double-stranded RNA in two lengths of about 25 bases. So in this work, the researchers describe a front view of the structure as looking like an X on the handle, and this is a domain that is known to bind a small RNA product, and on the blade and there is a domain that is able to cleave the RNA. So between the clamp and the cleaver is a flat surface region that carries a positive electric charge. So the researchers propose that this flat region binds to the negatively charged double-stranded RNA like a biological velcro, enabling dicer to measure out and snip specified lengths of siRNA. When you put the clamp, the flat area and the cleaver together, you get a pretty good idea as to how dicer works. So the research team is now using this structural model to design experiments that might reveal the triggers dicer into action. In addition, one size does not fit all for dicer. Different forms of the dicer enzyme are known to produce different lengths of siRNA, ranging from 21 to 30 base pairs in length or longer. Having identified the flat surface positively charged region in dicer as the ruler portion of the enzyme, the researchers speculate that it may be possible to alter the length of a long connected helix within its domain to change the lengths of the resulting siRNA products. So researchers would like to see what happens when you take a natural dicer and change the length of its helix. So RNA interference, genetic process in which targeted genes can be turned off. So necessary step in many fundamental biological events like genome rearrangement, stem cell differentiation, brain development, viral defense, etc. Can infer a gene's function by observing effects of switching it off. Can also be put to practical use, treating cancer, improving crop pest resistance. So dicer enzyme known to play a key role in RNA interference. Details of how it functions have remained unclear yet. Remain unclear yet. Okay. Now, if you want to talk about the mechanisms and applications of RNA interference, it's a beautiful poster I found online. We'll go through this. So obviously this is a poster, very beautifully made. Sorry. Yeah. Okay. So the revolutionary finding of RNA interference resulted when plant scientists in the US and the Netherlands tried to produce petunia plants with improved flower colors by introducing additional copies of a gene encoding a key enzyme for flower pigmentation into petunia plants. So when the scientists ended up with fully or partially white flowers, they discovered that both types of genes, the endogenous and the newly introduced genes, had been turned off. A few years later, plant virologists made a similar observation, and in their research they surprising observation that plants carrying only short regions of viral RNA sequences not coding for any viral protein showed enhanced tolerance or even resistance against viral infection. So they concluded that viral RNA produced by transgenes can also attack viral infection, right? So they concluded that viral RNA produced by can also attack incoming viruses and stop them from multiplying and spreading throughout the plant. So they call this phenomenon virus-induced gene silencing or simply VIGS. So these phenomena are collectively called post-transcriptional gene silencing, and many laboratories around the world search for the occurrence of this phenomenon in other organisms. Now, scientists A. Fire and C. Mello injected double-stranded RNA into C. elegans and noticed a potential gene silencing event. They coined the term RNA interference. So you see this is schematic of RNA interference. It's a mechanism basically. So what happens? Introduction of the double-stranded RNA triggers the RNA interference pathway. Dicer, which is cytoplasmic nucleus, leaves the dsRNA and which produces this siRNA, short interfering RNA, which then unwinds and assembles into RISC, RNA-induced silencing complex or RISC over here. Antisense siRNA strand then guides the RISC to the complementary RNA molecule. RISC then cleaves the mRNA. This leads to specific gene silencing. Now, since the mammalian cells mount a potent antiviral response upon introduction of dsRNA longer than 30 base pairs, researchers transfect cells with 21-23 base pairs siRNAs, thereby inducing RNA in these systems without eliciting an antiviral response. So what can be the applications? So RNA interference can be used as a tool for determining the gene function. So according to the central dogma of molecular biology, DNA is transcribed into RNA, which is then translated into protein. Now, proteins and some RNAs are the functional components within a cell. Thus, the ability to selectively destroy RNA allows researchers to effectively repress gene expression. Now, previous methods of studying gene function have involved manipulation of DNA. A typical experiment required the generation of random mutants by exposing a group of organisms to mutagens or through the use of DNA inserts. So the few that expressed a phenotypic change relevant to a given study were then selected and the genetic location of the sequence change would be determined. The advent of RNA interference-based gene silencing along with its use of genome sequence information has dramatically changed how these tests are done. RNA interference is a form of reverse genetics, meaning researchers can systematically pick genes rather than beginning with mutants and then searching for the genes affected. So one major advantage of this method is that the genetic location of any given mutation is predetermined, which removes a large portion of previous labor. So additionally, all genes can be analyzed if a gene is vital a mutant might not survive long enough to be noticed, let alone be studied. So with RNA interference, researchers can pick a chromosome and systematically knock out genes as they appear sequentially. Now, various methods exist for inserting dsRNA into target organisms. One method is direct injection, which, though currently used for medical applications, is not the best option for high throughput research. Viruses can also inject an appropriate duplex. There are two ways of expressing dsRNA in bacterial vectors: by encoding a hairpin structure or by use of a dual promoter that expresses both the sense and the antisense strand. So, in an experiment conducted on C. elegans in 2000, dual promoters were inserted into E. coli plasmids. The worms that ate these bacteria contained the dsRNA for several days, and it was carried on to their progeny that were produced within this time period. The embryos were taped as they developed, and any changes relative to wild type phenotype were recorded. So, macular degeneration is when the protein vascular endothelial growth factor or VEGF is overproduced in the eye. An excess of, I'll not go into the details of macular degeneration. So this was the basic. Okay. So, what is transcriptional gene silencing and post-transcriptional? In case of transcriptional, the promoters are silenced. In the other case, the promoters are active. The genes are hypermethylated in promoter region, where the genes are hypermethylated in coding region in case of PTGS. And this is just simply termed as RNA interference. Now, what are the other names of post-transcriptional gene silencing? Gene silencing, RNA silencing, RNA interference in certain fungi, quelling. So, the ability of exogenous double-stranded DNA, RNA, to suppress the expression of the gene which corresponds to the dsRNA sequence is called RNA interference. Introduction of transgenes homologous to endogenous genes often resulted in plants with both genes suppressed, called co-suppression, resulted in degradation of the endogenous and the transgene mRNA. Now, in 1995, injection of either antisense or sense RNAs in the germline of C. elegans was equally effective. Extension of above experiments, combination of sense and antisense RNA was 10 times more effective than single-strand RNA. So, dsRNA needs to be directed against an exon, not an intron, in order to be effective. Okay. Exon will be the coding part. Homology of the dsRNA and the target gene mRNA is required. Targeted mRNA is lost, degraded of the RNA interference. The effect is non-stoichiometric. Small amounts of dsRNA can wipe out an excess of mRNA. Okay? And sRNA does not work as well as dsRNA. So, double-stranded RNAs are produced by transcription of inverted repeats, viral replication and transcription of RNA by RNA-dependent RNA polymerases. Double-stranded RNA triggers cleavage of homologous mRNA. PTGS defective plants are more sensitive to infection by RNA viruses. In RNA interference, defective nematodes, transposons are much more active. So, RNA interference can be induced by, you see, this is a schematic. Okay, so cultured cells are taken, dsRNA is transfected or put inside the cells. Okay, and then the transfect plasmid is expressed in the mRNA precursor like stem loop. So the overexpression of dsRNA happens from the transgene to trigger cross suppression. Okay, so dsRNA is polyinterfering RNA, RNA induced risk or silencing complex, homologous mRNA target recognition, target cleavage. We have already talked about dicer in our previous class. It's basically double-stranded RNA triggers processed into siRNAs by enzyme RNA-C3 family, specifically dicer family, possessive enzyme, node larger intermediates. Dicer family proteins are ATP-dependent nucleases. These proteins contain an amino-terminal helicase domain, dual RNA SC3 domains in the carboxyl terminal segment, and dsRNA binding motifs. They can also contain a PAS domain, which is thought to be important for the protein-protein interaction. Dicer homologs exist in many organisms, including C. elegans, Drosophila, yeast, and humans. Loss of dicer means loss of silencing processing in vitro, developmental consequence in Drosophila and C. elegans. Now, RISC is a large RNA multi-protein complex which triggers the mRNA degradation in response to siRNA. Some components have been defined by genetics, but function is unknown. Unwinding of double-stranded siRNA, ribonuclease component cleaves mRNA, amplification of silencing signal, etc. During dsRNA cleavage, different RNA classes are produced: siRNA, miRNA, sTRNA, etc. siRNA is small interfering RNAs that have an integral role in the phenomenon of RNA interference, a form of post-translational gene silencing. RNA interference leads 21-25 nucleotide fragments which bind to the complementary portion of the target mRNA and tag it for degradation. A single base pair difference between the siRNA template and the target mRNA is enough to block the process. Small temporal RNA is derived from 17 nucleotide ssRNA which forms the stem loop, etc. Role the temporal regulation of C. elegans development, preventing translation of the target mRNAs by binding to the target's complementary 3' UTR. 15% of these mRNAs were observed with 1-2 mismatches across worm, fly, and mammalian genomes. The expression pattern can vary. Some are expressed in all cells and at all developmental stages, and others have more restricted spatial and temporal expression pattern. Okay. So why is it important? Because most widely held view is that RNA interference evolved to protect the genome from viruses. Recently, very small RNAs have been discovered in several eukaryotes that regulate developmentally. Other large RNAs may be used for RNA interference mechanism, maybe defense. So, what are the applications of recent applications of RNA interference? Modulation of HIV, one application. Potent and specific inhibition of human HIV virus. Selective silencing of viral gene expression in HPV positive human cervical carcinoma cells. RNA interference in adult mice. Successful inactivation of endogenous of 3x4 CMOS genes in mouse. Pre-implantation embryos. Okay. So what are the feature improvements of RNA interference applications? In vitro synthesis of siRNAs, U6 RNA promoter based plasmids, direction of longer dsRNA, potentially useful creation of siRNA vectors, establishment of an inducible siRNA system, establishment of retroviral siRNA vectors. So these are the conclusions. It's powerful for analyzing unknown genes and sequence genomes. Efforts are being undertaken to target every human gene. We talked about gene therapy, downregulation of genes, mutated alleles, knockout of genes for required for cell proliferation, knockout of genes including key structural proteins, etc. So, coming back to the question for today, RNA interference was discovered in C. elegans. If you have any doubt, you can ask me now. We'll wait for 4-5 minutes and we'll move on to the next question. Okay, so there are no questions. We move on to the last question for today. Genomic imprinting is not seen in all living organisms. True or false? I'll give you a few minutes to think about this and answer, then we'll discuss. Anyone? Okay, so the answer will be true. Okay, so we'll now learn what is genomic imprinting first. So genomic imprinting is basically a process that marks genes based on their parental origin, right? It's a type of epigenetic gene regulation that can lead to human genetic disorders. So, how it works is that during egg and sperm formation, chemical tags called methyl groups are added to certain segments of DNA. Now, these tags identify which copy of a gene came from the mother and which came from the father. So, the tag silences the copy of the gene that came from one parent while turning on the copy from the other parent. Now, how is it significant? Genomic imprinting is different from classical genetics because the parental complement of imprinted genes are not equivalent. So many imprinted genes play important roles for development. So abnormal expression of these genes can result in numerous human genetic disorders, including carcinogenesis. Okay, so this is genomic imprinting. So, two alleles are inherited from mother and father in higher eukaryotes, including humans. So, both alleles are usually functional for the majority of the genes. However, one of them is sometimes switched off or stamped, right? So, this allele or a separate gene is defined as imprinted in offspring. Imprinting is the gene silencing in the offspring, though the same gene obtained from the other parent remains expressed. Okay. So genomic imprinting is inheritance lying beyond the Mendelian borders. A range of inherited diseases and human development features violate the Mendelian law of inheritance, and this way of inheriting is called epigenetics. Okay. So epigenetics shows that gene expression can change in more complex way than modifications of the DNA sequence. Epigenetics considers the environmental impact on the gametes prior to the fertilization. So the gene and genome imprinting mechanism are not still completely clear. It involves epigenetic modifications which are erased and then reset during the egg and sperm formation, and it's a process of the gene silencing reached through DNA methylation. So the repressed allele is methylated, whereas the active allele remains unmethylated, and the most well-studied conditions belong to a Prader-Willi syndrome and an Engelmann syndrome. So these two syndromes are supposed to arise from genome imprinting or other gene-related errors involving the long arm genes of chromosome 15. So genomic imprints change in a regular way during the life cycle of the organism. Imprints are set up during the development of the gametes or the germ cells into sperm or eggs. After fertilization, they are maintained as duplicated but separate chromosomes in the developing organism. So in the haploid gametes, germ cells of the developing organism, of the new organism, imprints are erased at an early stage, so then they are re-established at a later stage of the gamete development to finish the GI cycle. So in diploid somatic cells, imprints are maintained and modified during development too. So the imprints that are introduced in the parental eggs and sperm or the germ lines remain in the early embryo and fully matured zygote during differentiation. These imprints control the genome, so their reading means that genes are expressed differently due to the chromatin methylation. So, in result of genome imprinting, the allelic expression is differential. It refers the expression from one parental locus over the other. Okay, so scattering CPG islets or diets in the early cleavage embryo point to a continuous partial loss of methylation. It is then repaid by selective de novo methylation. A combination of passive and active demethylation events, counteracted by de novo methylation, are involved in the distinct reprogramming dynamics of DNA methylomes in the zygote, so early embryo and primordial germ cells, primary cells, eggs and sperms. If you talk about the Prader-Willi syndrome, it's characterized by abnormal feeding and appetite and learning disability. The patients may also develop a severe affective psychotic illness similar to bipolar disorder. So this includes loss of antisense transcripts, which represses the expression of UBE3A, which encodes E6AP. This is acidic protein, ubiquitin ligase from the paternal chromosome. So the paternal copy of this gene, which is normally expressed from the maternal chromosome, becomes reactivated, leading to increased dosage. What we know about Angelman syndrome? It is a neurodevelopmental disorder characterized by severe cognitive disability, motor dysfunction, speech impairment, hyperactive behavior, and frequent seizures. So, is caused by disruption of the maternally expressed and paternally imprinted UBE3A, which encodes an E3 ubiquitin ligase. So, unlike Angelman and Prader-Willi, the 15q11q13 imprinting chromatin region has also been linked to other non-syndromic neuropsychiatric illnesses. For instance, maternal duplication of the interval is associated with the incidence of autism. So, throughout the years, extensive efforts have been made to characterize the epigenetic marks underlying genome imprinting in animals and plants. The data collected to date stress that genome imprinting cannot be solely explained by DNA methylation or demethylation for the imprinting of all genes. At present, new models explaining genome imprinting regulation in plants are advanced as functionally independent of parental DNA methylation, asymmetric in the genome imprinting setting. We will not go into much details. So, concluding, it's important process of inheritance that plays a significant role in the future genetic and epigenetic studies. It's a complex process, the core of which is based on DNA methylation in alleles of chromosomes. Numerous external factors influence DNA methylation, which may cause the disease onset or its progression, namely in offspring neurodevelopment and autism spectrum disorder. And it's a rather rare phenomenon in humans. The majority of genes isn't imprinted. So modern studies were done with mice or plants. So these data should be completed. So the precise mechanisms underlying epigenetic gene control in the pathogenesis of several diseases remain unclear, but there are findings proving that the progression of such diseases can be eliminated or altered by modulating epigenetic programs. Okay, so the data summarized to date show that genome imprinting cannot be explained only by DNA methylation or demethylation as the main factor of the imprinting for all genes. New models explaining the genome imprinting regulation in plants have been elaborated as function besides the parental DNA methylation asymmetries in the genome imprinting setting. So, coming back to the question that we started with, this is true. It's not seen in all organisms, all living organisms. So if you have any doubts regarding this question, you can ask me today, or we'll stop or conclude the session for today and meet again next Monday. So any questions? I'll wait for 2-3 minutes. If you have any questions, you can ask me now. Okay, so if you don't have any more questions, we can conclude the session for today. And we'll meet again next Monday, same time. Thank you so much. Thank you, everyone. I'll stop the session.
Back to school time.